Today we'll be dealing on the topic GURT. GURT is the name suggests genetic use restriction technology. So this technology restricts the genetical use, not all the sorts of genetical use, but the unauthorized genetical use. For example, if a company has produced a genetically modified crop, like rice, genetically modified rice, then the company sells them to the farmers, but it does inserts this GURT technology in that plant so that every other generation the farmer need to buy the seed from the same company. The farmers cannot save the seed for the further generation so that the company can get benefit. So this is majorly termed as Terminator technology, and it is facing a lot of criticism worldwide. Well, there are two types of GURT basically. They are variety GURT and Terminator GURT, T-GURT, variety GURT, V-GURT, or variety GURT and T-GURT, or trait associated GURT. The Terminator technology that we have been hearing all around is variety GURT or V-GURT. We'll be dealing about Terminator technology and V-GURT in detail. But T-GURT is not totally a Terminator technology, but it does have some mechanism, but it is quite advantageous and it is quite flexible in comparison to V-GURT. So what are they? What are the terminology? What are the genetic mechanism behind them? We'll be dealing about them. Okay. Now, what are the major components of a? Okay. What are the major components of GURT technology? Well, it is GURT, genetical. We are all dealing with the genetical resources. Genetical resources means we have to deal with the gene, and what is there in the gene of the genetically modified plants or crops that makes them terminated, like terminator seeds, how the terminator seeds produced, and what are the major components behind them. So major components, the first one is the repressor protein. This is the repressor protein coding gene. The other one is Cre recombinase coding sequence, and finally the toxin gene. We have all Cre recombinase and LOX, relationship between Cre recombinase and LOX. If not, we can know about them and come back. Cre recombinase always clips away or cuts the LOX P sites and joins the other two adjacent sites with each other. So Cre recombinase is an enzyme playing a role in the entire mechanism of production of the sterile cells. So how do they work? Let us see here. With every gene, for every gene there is a kind of promoter, like for repressor gene there is this promoter, for Cre recombinase there is a promoter, and for toxin gene also there is a promoter. This LEA is late embryogenesis abundance. Why is it called late embryogenesis abundance? Because these promoters are expressed only in the late embryogenesis or we can say when it's the seedling stage or embryogenesis stage, only they are activated. So what are the components? Like repressor protein has a promoter, this Cre recombinase have a 35S promoter, and this toxin has a promoter. So there's an interrelation. What it does is repressor protein binds to the promoter of Cre recombinase and it cannot be expressed, and if it cannot be expressed, this LOX P component or DNA that is present in between the promoter and toxin, this LOX P is inhibiting the expression of a toxin because the promoter is separated from the gene of expression. So until and unless this gene of this LOX P gene is removed by the Cre LOX P, toxin cannot be produced. So this is the basic mechanism. So if and only if Cre recombinase is expressed, LOX P is removed and toxin is expressed. Otherwise, if Cre is not expressed, LOX P is not removed and this toxin is not expressed. So this is what we have to understand. So how the seeds are produced. So if a company has to sell a seed for the first generation, the seeds must be fertile, because when a farmer buys the seed from a company, they sow it in the farmer food productivity. Like if the farmers buy a rice seed, they have to plant the rice seeds to get rice. Okay, now the rice seeds they have grown, and after the second generation rice seeds, if they try to say they cannot be grown into fertile, they cannot be grown into plants, but the first generation seeds that are sold by the company should be fertile, otherwise the farmers won't buy them. So how are the fertile seeds for the first generation seeds turned fertile? Okay, what they do is this, I have already told, this repressor protein that is present in the genetically modified seeds are constantly active, and they are constantly produced. This repressor proteins produced have the tendency of binding to the promoter region of Cre recombinase enzyme. So if the repressor protein binds to the promoter of this Cre recombinase, the Cre recombinase cannot be expressed. So lacking the activity of Cre recombinase, the LOX P gene is not clipped out, and it still remains in between the promoter of the toxin gene. So toxin gene here cannot be expressed. The first generation, the seeds are produced with the repressor. That is why the toxin is not produced and the seeds are viable. These seeds are sold to the farmers, and they are grown in the field, and they grow to give the plant life. Now in the second generation, how the seeds turn to be sterile. What the company have their own self motive that the farmers need to buy the seeds. For every time they use the seeds, they have to buy the seeds from the company. So what they do is, before selling the seeds to the farmer, they treat the seeds with the tetracycline. Tetracycline acts as an inducer here. So they have sold the fertile seeds to the farmers, and those seeds are treated with tetracycline, right? Now this tetracycline go and binds to the repressor protein that were present here, right? The repressor protein are bound by the tetracycline. Thus the repressor protein that when bound by the tetracycline are not available to the promoter of the pre-recombinase enzyme. This is why the pre-recombinase get expressed. So the farmers sow the seeds, the plant starts growing. As they start growing, and they turn bigger and bigger, and the pre-recombinase also starts accumulating. Pre-recombinase starts accumulating, and they cut the LOX-P site of this toxin gene. And as the plant grows and reaches to the late embryogenesis stage, this LEA and toxin protein gene have also come together. So the toxin protein can be expressed, and when the plant grows and reaches late embryogenesis stage, this toxin protein is expressed abundantly, and the toxin gets accumulated in the embryos of the second generation seeds. So the farmer have grown the seeds, but all the seeds have accumulated the toxin that has resulted in making the seeds non-viable. So for the second generation, if the farmer try to plant those second generation seeds to get the next generation, they cannot because they have already been sterile. So this is the basic mechanism behind the V-GURT or variety GURT. And what is there in T-GURT? T-GURT or trait associated GURT. In T-GURT, there is an option